Good morning, viewer. Welcome to our English version. Yes, really a breaking news for our viewer. Researchers in Leiden have, for the first time, observed how a specialized enzyme helps bacteria stay alive when oxygen levels are low, and how that processes can be blocked. The study published in Science Advances opens up new possibilities for targeted antibiotics. It really exists, a secret trick that allows bacteria to survive with very little oxygen. And this also applies to bacteria that can make us sick. Like us, these bacteria need oxygen to survive, says PhD candidate Tizin van der Valden. But unlike humans, they have a special enzyme called cytochrome BD that allows them to keep producing energy even when oxygen levels are very low. Because the enzyme is so important for bacterial survival, and it is a promising target for new antibiotics, including potential treatments for tuberculosis. For years, scientists have been trying to understand exactly how cytochrome BD works and how existing inhibitors block the enzyme. But studying it has been difficult because the relevant part of the enzyme is highly flexible and constantly moving, so making it hard to capture clearly. We needed to find a way to stabilize it, Van der Velden explained. The breakthrough came thanks to the new glacios microscope. Using it, the researchers were able to study the enzyme at atomic level. They carried out the work in Escherichia coli, a bacterium commonly used in laboratory research. Previously, the team used the advanced cryos microscope at the National Imaging Center NECEN, but that microscope is often booked solid, so you only get one or two chances to collect data, and everything has to work immediately, says supervisor Lars Juken. And he also expressed, now we could repeat experiments, adjust our methods, and improve the processes step by step. In the end, the key turned out to be changing the way the enzyme was removed from the bacterium's fatty cell membrane, a necessary step before it can be studied. Chemists normally use a fairly strong detergent for this, but the researchers choose a milder alternative. So that made a major difference. With this detergent, the enzyme came out as dimers, pairs of identical proteins that remain attached to each other, says van der Velden. With stronger detergents, the enzyme probably fell apart, leaving only unstable single proteins behind. So the dimers turned out to be more stable and less mobile, allowing the researchers to clearly examine the flexible part of the enzyme for the first time. So that finally allowed us to see how this enzyme works, says Van der Velden. No one had managed that before. Zeucon adds. Bow down, ma'am. BNS exclusive report, FTB English, Delhi, Bharat.